Welcome to Bioterrorism, Chapter 30 in the Mahon textbook. Some terms to know. Bioterrorism is the use of bacteria, viruses, fungi, or toxins to injure people, animals, or crops to cause civil and economic unrest. And biological warfare is the use of bacteria, viruses, fungi, or toxins to injure people, animals, or crops to gain military advantage. So that is the difference. So questions to answer. Who will be the first to suspect a covert bioterrorism attack? And the answer is microbiologists and physicians, because you'll see different things coming in the lab, or people with different ailments, or a lot of people with the same ailments showing up into the hospital or healthcare system. But what document prohibits the use of biological agents during war? And the answer is the Geneva Protocol. So we need to talk about the biosafety levels, or the BSL. And the BSL classification system was developed in 1999, and it is based on a microorganism's disease potential. It's a combination of standard procedures and techniques, safety equipment and facilities designed to minimize the exposure of workers, who are us, and the environment, also our environment, to infectious agents. Each increase in level, in BSL level and biosafety level, indicates more precautions and work controls. BSL 1 is the lowest risk, and those are organisms that do not normally cause disease. There are minimal safety procedures and equipment that are needed, and an example of this organism is Bacillus subtilis. And if you remember, that's the one that we use as a quality control organism in our autoclave systems. BSL-2 organisms cause human disease but are not transmitted among hosts. Work should be performed under a Class 2 hood. Examples of Class 2 biohazard safety level organisms: hepatitis B virus and salmonella. The next BSL is 3. Organisms transmitted by a respiratory route. These cause serious disease, and they require the use of a separate air ventilation system. An example would be Mycobacterium tuberculosis. So tuberculosis is a BSL-3 organism. BSL-4 organisms are transmitted by a respiratory route. They cause a high risk of disease, and very strict precautions, including containment suits, are required to work with BSL-4 organisms. There is no treatment or vaccine, and an example would be Ebola. Now we have, in addition to the BSL levels, we have 3 categories for public health preparedness. And this is different, and so we're going to talk about Bacillus anthracis in a minute. So public health preparedness, category A, include agents of greatest impact, and examples of those would be Bacillus anthracis and Yersinia pestis. Class B are those that are easily disseminated, like Brucella and E. coli 0157H7. And then Class C are the most dangerous. They are the emerging pathogens that can be disseminated to the masses, and examples would be multi-drug resistant Mycobacterium tuberculosis and something like the hantavirus. So special notes about anthrax. Bacillus anthracis, while it's a BSL-2 in the clinical lab, it's a category A in regard to bioterrorism threat. So remember that those don't have to be the same level. Inhalation anthrax is the most deadly, and it takes about 8,000 spores to initiate disease. And also, in the 1970s, there was an accidental release of anthrax spores in Sverdlovsk, Russia. So if you have some free time, that is an interesting story to read. So there are some things that need to be considered when we're talking about bioterrorism. And biological weapons usually enter a host via respiratory tract, GI tract, skin, or mucous membranes. So primarily we're concerned about anything that can be aerosolized And thank goodness, food and water sources are usually not used. However, in the United States, in Oregon, in 1984, someone sprayed liquid salmonella solution on a salad bar, and about 750 people were infected. That's 750 people were infected on purpose. Nice. Now we're going to talk about the Laboratory Response Network. It was established in 1999 by the CDC, the Centers for Disease Control, the FBI, and the United States Army Medical Research Institute of Infectious Disease, or USAMRID. And so here are all the logos right there. The goal was to link state and local labs with advanced capacity to clinical, military, veterinarian, agriculture, water, and food testing labs, to link all those and decentralize the testing capabilities. And we've learned in 2020 how important that is, to decentralize testing capabilities. The laboratory response structure goes like this. Level 1 are the sentinel labs. Those are the ones on the front lines. The role played by the sentinel labs is to rapidly detect potential threats, rule in or out suspicious isolates, not to go through the full identification process, and provide assistance on proper collection and processing and handling of samples. Community hospitals with microbiology capabilities are the sentinel labs, and these have BSL-2 capabilities, these labs. The next level is the reference lab. Their role is to offer confirmatory testing for bioterrorist threats, culture, identify, and examples of these reference labs would be the state public health labs and Department of Defense at the state level. The national level, the role of the national labs would be to perform all reference lab tests and forensics testing. So that means they are going to probably genetically fingerprint the organisms to see how they're being passed, and if it is the same strain from the same point of origin. They will be the ones to decide that there is a definite ID and biothreat agent, and examples of these are the CDC, USAMRID, and the National Research Medical Center. And these labs have the BSL-4 capability. So some test questions might be, which facility is designated as a sentinel lab? And remember, those are our community hospital labs that have a microbiology capability, and those are the places that you will be doing your clinical rotations. So what indicators do we have that there might be a potential biocrime event? So if we start seeing a disease that's usual or does not occur naturally in a given area, if you have multiple diseases in the same patients, higher than average morbidity and mortality, massive point source outbreak, aerosol route of infection, or an illness limited to located or certain local or geographic location, dead animals, or the absence of a competent natural vector in the area of an outbreak. Agents of bioterrorism that we may hear about or come into contact with: Bacillus anthracis, Yersinia pestis, Francisella tularensis, Brucella species, Burkholderia species, Coxiella burnetii, variola viruses, which causes smallpox, hemorrhagic fevers, ricin, which is a toxin, staph enterotoxins, and Clostridium botulinum toxins. So I've pulled up some pictures for you of what we might actually see in the lab. So for Bacillus anthracis, remember, these are going to be non-beta hemolytic, crusty, dry colonies that are non-motile. Close-up of the colony, you'll see medusa heads. Maybe they have a ground glass appearance, and also the colonies are very tenacious or tacky. So if you take your loop and touch it and lift it, you'll have just like a little pointy finger that sticks up, it has like a waxy consistency. For Yersinia pestis, you'll see the safety pin gram stain, and on blood agar, you will see a fried egg appearance and a non-lactose fermenter on McConkey. For Francisella tularensis, you're going to see like a really poor looking gram stain of pleomorphic, very tiny gram-negative coccobacillus, and you might see growth on BCYE, chocolate agar, sheep's blood agar, or cystine. So nothing really stands out with our regular media that we'll be culturing. For Brucella species, remember that we can get this through unpasteurized milk, and this grows on sheep blood agar and chocolate as very small non-hemolytic colonies, and the gram stain will reveal tiny gram-negative rods. For Burkholderia species, glanders is an infectious disease that's caused by Burkholderia mallei, and people can get the disease. Glanders is primarily a disease affecting horses, also donkeys and mules, and it can be seen in other mammals such as goats, dogs, and cats. Meloidiosis is also called Whitmore's disease, and it is caused by Burkholderia pseudomallei, and we have talked about this briefly in another chapter. Coxiella burnettii causes Q fever, named Quarry fever after an outbreak in 1935 in an Australian slaughterhouse. No cause was ever found. Smallpox is caused by the variola virus. Pustules penetrate deep into the tissue and they can leave scarring all over the body. Then we have the hemorrhagic fevers, most notably Ebola. We just had a case on United States soil in 2019. Those look pretty terrible. And then toxins, we have ricin. Ricin is one of the most poisonous naturally occurring substances known. It's from castor beans, and if you like to watch crime dramas, I'm sure you've seen a show or a place where a plot where they use ricin. Clostridium botulinum toxin is a neurotoxin, and while it can cause food poisoning, that is the stuff that they use in Botox to get rid of wrinkles. And that concludes our molecular microbiology chapter discussion on bioterrorism.